Hi, everyone. My name is Yuna, and I'm a PhD student in the Gerges Lab at Harvard. And today I want to talk about viruses in the deep sea, in a microbial mat, actually, where we find novel modes of host-virus interactions that seem to cross-cut taxonomic domains. So I'm interested in understanding host-virus interactions in natural environments. And what we know about microbes in nature is that a lot of these microbes exist in aggregates and often in forms of biofilms. And biofilms, as Alex talked about just now, often consist of substrate-bound and sessile microbes, are high in biomass and feature spatial organization and structure that are often maintained by EPS or extracellular polymeric substances. And we find microbial biofilms in marine environments, to gut lining, to often in microbial mats, which is a macro feature that we can see with our eyes. And another thing that's cool about microbes in nature is that microbes in natural environments seem to feature metabolic interdependence or syntrophy, and it seems to be a lot more prevalent than what we see and have studied so far. And it's been studied in gut as well as in marine environments where we find AMI and SRB consortia of archaea and bacteria working together to carry out complex metabolisms. So despite these complexities, host virus interactions in laboratories are studied in liquid homogeneous cultures, often consisting of single species. So we know quite little about host virus interactions in biofilms, especially in syntrophic biofilms. So we began with this question of how do viruses interact with hosts in biofilm communities that feature syntrophy? And we posited that with extended contact between syntrophic microbes and their viruses, and high density of phylogenetically diverse microbes, and limited dispersal of both microbes and viruses due to EPS matrix, might lead to a prevalence of interactions between viruses and non-host cells, or an emergence of broad host range viruses. So in order to study this, we went to one of the most extreme examples of syntrophic biofilm, which would be a deep sea microbial mat. So these hydrothermal mats take place or are found in this interface between cold oxidizing seawater and hot hydrothermal fluid rich sediments. So as the in this redox gradient, we find chemosynthetic life really thriving and forming these beautiful mats that are quite thick and fluffy and large as well. So these deep sea microbial mats or hydrothermal mats are a unique environment to probe understudy dynamics between metabolic linkages and host virus interactions. So we went to the Guaymas Basin, which is which is here in the spreading center. California is up here, Mexico is down here in Gulf of California. And we used an ROV to sample a decently large microbial mat that was about 10 meters wide. And we took a transect of push pores across this axis. And then what we find here is that this mat is spatially heterogeneous. And this is proxied by temperature, which ranges between 20 degrees to 55 degrees within this span of a few meters. And it's dominated by sulfur oxidizing Vegetoa of different morphologies, ranging from orange fluffy filamentous morphology to more granular and white filamentous morphology. And although I don't have the time to go into the details of the community structure, they feature both sample-specific and mat-wide viral and microbial communities. So we started off with 10 mat samples, and then we used shotgun metagenomic sequencing in order to bin microbial mags and viral mags from this 10 samples. And using the CRISPR arrays that were abundant in these microbial mags, we recovered the sequence spacers from the raw reads and then use spacer to protospacer matching in order to infer historical host virus interactions. And then we use the same samples in order to conduct Hi-C library sequencing. Hi-C essentially is when sequencing with, putting the sequencing chimeric reads where genomes from chromosomes that are in contact are cross-linked, and then using that chimeric reads in a computational way to infer in situ genome co-localizations. So using the MAGs and VMAGs that are bin from the metagenomic sequences, we were able to infer in situ host virus genome co-localizations or interactions. So what we're showing here is historical host virus interactions using CRISPR protospacer and spacer matches. And what we see here is a lot more nested than what we would typically expect from what we know about phages and viruses and their quite narrow host ranges or host specificities. And what we have here is these colorful nodes are host nodes or microbial mags, and the gray nodes are viral nodes. And when there's an edge between them, there is host spacer to protospacer matches, but at least two, but oftentimes more. So in this case, there's a lot more spacer to protospacer matches. And what's cool about this is that we see a virus that seem to interact with diverse hosts, but not just in different phyla, but also in different domains. So these are SRBs, so sulfur-reducing bacteria, and AMNI, Anarchaea. And we see this pattern quite pronounced in pairs or groups of microbes that seem to be undergoing syntrophic metabolisms. And we see the same pattern with SOB and SRB. And the syntrophy between SOB and SRB, so sulfur oxidizing bacteria and sulfate reducing bacteria, have been hypothesized by the field too. And now we're showing the Hi-C inference network. So to parse this network, we have viral genomes in the middle, and the size of the viral nodes will correspond to the viral Vmax size. And then microbial genomes are around in the circle. And when there is a edge between the viral node and a microbial node, the thicker edges represent higher level of Hi-C linkage, possibly representing primary host or even possible infection. So it seems that there are oftentimes main primary nodes or groups of hosts that a viral mag can or virus can infect, but there seems to be other interactions that are prevalent in this system too. And these red edges represent host virus interactions that have been previously identified using the CRISPR-based method in the network that I showed before. And what's cool about these networks also is that viral genome size seem to be correlated to host range, and in both CRISPR spacer-based method as well as Hi-C proximity ligation-based method. And I used the word host range in quotation marks here because how we think about host range in this study is quite different from, or it expands beyond what we traditionally think of as host range, which is infection-based, or it implies infection. So in order to explain this pattern that we've seen, we devised or we proposed a couple of models or few models to explain these observations. So in this microbial mat, we can possibly imagine a syntrophic pair of phylogenetically distant and metabolically interdependent microbes such as ANMI and SRB. And when there is a lysis event of a virus that's infecting this its primary host. Because of the EPS matrix, there is limited diffusion of viral particles and also viral DNA. And when there is a lot of viruses accumulating in this system, there can be promiscuous viral absorption as well as entry without infection. And this kind of sort of promiscuity in absorption has been seen in through other methods such as epic PCR recently. And this would result in, even though this virus is not able to necessarily infect or carry out a full infection in this partner or syntrophic partner, this would still result in a CRISPR gain event, CRISPR spacer gain event by this CRISPR array or system reacting to responding to this viral DNA. And we can also imagine a scenario where a viral DNA that's accumulating getting taken up by naturally by the syntrophic partner as the viral DNA concentration increases. We can also imagine a scenario where there is a contact-based transfer of DNA or viral particles between the syntrophic partners. And this kind of conjugal transfer of DNA across large distances has been hypothesized and observed, although it requires a lot more verification. But what's cool about both of these scenarios is that it's going to require a CRISPR-mediated defense by the syntrophic partner, which would lead to an increased community-wide adaptive immunity, which kind of suggests that there might be a collaboration or sharing of the load of immunity between the syntrophic partners. We can also imagine a scenario where there is host switching, and these mats being a very like a hotspot for host switching, where when they're in high proximity with each other over a long time, host will evolve against the virus, and then the virus might find its next closest physically close host, which would be its syntrophic partner. So this can happen over time, or it can happen also through a broad host range model where there is bona fide infection happening. Although this might be more of the case for more closely related hosts, not necessarily for bacteria and archaea, considering their different physiologies. So, so what we're trying to get at here is that we think that host virus interactions may be a lot more nuanced than previously thought. So what is thought to be pretty narrow in host range or binary between lytic and lysogenic, we think that there may be this whole array of ways in which viruses and microbes can interact with each other. So you might ask, why do these nuanced interactions even matter? Because in the end, they don't necessarily kill the host. Well, they still matter because these viral genomes may still shuttle important genes across large phylogenetic distances. And although I don't have the time to go into detail, these viral genomes do indeed encode many auxiliary metabolic genes. And secondly, foreign DNA still places selective pressure on microbial defense machineries. And when we did the selection analysis on the microbial and viral genes, the microbial genes undergoing positive selection are often associated with defense. And lastly, which I think is the coolest part of the story, is that this provides a possible mechanism for shared immunity in a microbial community beyond within population pan-immunity. So it adds another layer to microbial syntrophy and symbiosis to extend beyond metabolism into immunity too. So to end with, the take-home message here is that we think that host virus interactions may be a lot more nuanced in nature, and these broad range interactions still have important ecological and evolutionary implications, and they require a lot more further investigation. And with that, I would like to thank my PI and the lab, the Gerges lab, as well as our collaborators, Simon and Clement at JGI. And of course, I'd like to thank the crew members and scientists who helped us collect these precious samples in the Guaymas, and the funding sources too. Thank you.
All right, I think we have time for one question. I see some all the way in the back.
Hi. So looking at the CRISPR spacer matches, it looks like I think you commented on this too. It was some of the nodes were hitting proteobacteria and archaea at the same time. How worried are you that the spacers are misassigned to the host?
Yeah, so that's definitely one of the things that we were worried about. So we discard any spacers, or we discard. So when we are assigning CRISPR arrays to the host, we only consider CRISPR arrays that are unique to the host, or CRISPR repeats that are unique to the host. So there were some that were kind of ambiguous, and we discard those. And also we only consider very high confidence viral context, and also high quality mags or complete mags. And we were also kind of concerned that these spacer matches were spurious. So we only consider those that have only less than 2 mismatches across 35 base pairs. And then the edges that we see here are when there's at least 2 spacer matches. So whatever is just one is not gonna make it to the network. Yeah. Short read, yeah, that's right.